Thanks a lot, Hank. It's a really pleasant duty that I have now to introduce our first speaker, Kristi Anseth from the University of Colorado, who I've known since she took up her first faculty position in 1996. She received her PhD also from the University of Colorado in '94, and then did a 2-year postdoc with Bob Langer, and came back to Colorado in '96, and is, as it's mentioned in the program, a year 2000 Teacher Scholar awardee. She is now the Tisone Distinguished Professor of Chemical and Biological Engineering Surgery and Howard Hughes Medical Institute investigator. Her work has really been phenomenally productive and influential in the development of biomaterials, scaffolds, hydrogels with highly controlled architectures, chemistry for 3D structures, for 3D cell culture, tissue engineering, biological assays. Kristi has always been at the forefront of applying new chemistries to the biomaterials field, and this is how her influence has played out. Building on her Teacher Scholar Award in 2000, she's now been elected to all 3 national academies, engineering, science, and medicine, is the author of well over 300 publications, and is going to talk to us about cellular control in a couple of clicks.
All right, thank you for the kind introduction. And before I begin, what I would say is that I started my career, I trained in chemical engineering. And my PhD was in this area of polymerization reactions and making different types of polymeric materials with controlled properties. And I hadn't had a biology class before, since high school, when I graduated in '94. And I jumped in and did a postdoc with Bob Langer and became very interested in how one could use different types of reactions, in particular photochemical reactions, to design new types of materials and chemistries that could be done in the presence of living systems. And at the time, the field of tissue engineering, regenerative medicine was burgeoning, and I was trying to bravely write many proposals in this area where many people thought that some of the ideas were maybe a little naive, maybe trying to push too far and crossing different boundaries, interacting with biologists, medical clinicians, chemists, and engineers. And so when that letter came, we still had letters back then, from the Dreyfus Foundation that I was selected to receive the Teacher Scholar Award, it meant a lot to me, and it had a big impact on my career. It gave me some confidence to pursue some of these ideas, but more importantly, some seed funds so we could get some of those key experiments done and invest in people in my lab and give them the freedom and ability to be brave in exploring this frontier. So I wanted to say a special thank you to the foundation. I think it's impacted many of us in our careers. So what I'd like to tell you about today is walk you through a little bit about where we've landed 15 years later from when I got that letter. And we've been very interested in this area where of cellular therapeutics. So the idea that was burgeoning at the time was using one's own cells to get the body to heal itself when processes had gone awry. And of course now we have many different sources of cells that one can use in pursuit of that goal. And perhaps the ideas weren't necessarily so new in the first place, in that blood transfusions have been around for decades and saving many people's lives. Some of you may have known people that have had bone marrow transplants to treat certain types of cancers. More recently, the Edmonton protocol takes a donor pancreas and gets the islets that make insulin And they'll transplant those islets into patients that have diabetes that's very severe and can't be regulated by insulin injections. So another cell-based therapy. We also now have products where one can take your own cartilage cells, your own chondrocytes, and inject them into surfaces of joints that have damaged cartilage tissue and regenerate that tissue in a controlled fashion. So we're starting to see more and more of this expanding in the field, and in fact, if you look at the FDA's website, you'll find tens of thousands of clinical trials of ways to inject cells into patients to try to alleviate debilitating symptoms of neurodegenerative diseases like Parkinson's, or into patients' hearts after they've had a heart attack with the hopes of trying to repair or regenerate some of that last cardiac muscle. So in that context, though, some of the motivation and themes in our work are that while these cell-based therapies we think are going to continue to grow and we're going to learn more, and there'll be more medical products, many times these treatments are suboptimal because when you inject those cells, oftentimes only a few percentage of them survive. It's called engraftment, so they integrate with the tissue and survive and function. And that's a tremendously low amount, and that leads to many suboptimal types of treatments. And so one can envision that our bodies are much more than 100 trillion cells, but instead they're embedded in a tissue, in a material environment. So one can think of that as complementary to all the advancements in cell biology and stem cell biology. We need a corresponding advancement in ways of new types of materials that could be used to deliver those cells or expand them in vitro or design new models of tissue disease. And so from that perspective, what new materials, chemistry, and engineering are needed to enable this? And so the way that we tend to think about this is we think about these cells being embedded in a microenvironment or a niche. And if one can embed them in these cells in 3 dimensions, those scaffolds can serve as a framework upon which those cells can regenerate and regrow tissue structures. Here I'm showing a picture of cartilage, is what some of our early work was focused on, was regenerating cartilage. But the field became stuck a little bit. We were able to regenerate tissues that had certain mechanical properties, like cartilage that's a low friction cushioning surface on moving joints. We were able to regenerate skin that could be useful for patients that had burns over large areas of their body, but mostly the barrier properties of that skin. We couldn't regenerate and innervate it or the hair follicles and whatnot. And the challenges become more difficult. We still don't know enough to get a small injury to the spinal cord, a very small injury, to regrow and integrate across that gap. We're very far away from being able to regenerate something like a small unit of your liver or your kidney that could be useful for patients that had failure of those organs. So those types of tissues are complex, and they have many different chemistry functions, metabolic functions associated with them. So this is where I think material chemistry and polymer chemistry and biomaterial science can play a big role, because we need to learn more about how do these cells get information from that surrounding environment, and how does that outside information get translated to the nucleus of the cell and control something important. If I want those cells to grow in number, if they want them to secrete and make a tissue, if I'd like them to differentiate into a specialty cell type. And it's not just about learning which signals are needed, but in what context and on what time scale. So this is the space that we're very interested in. And there are many different types of materials one can use for this type of application and answering these types of questions. And so one that my group has been very interested in is a class called hydrogel systems. And here I'm just showing a macroscopic picture of a hydrogel. From a very simple sense, if you could see the molecular level details of this hydrogel, it's composed of long macromolecular chains that would normally dissolve in water, but because of interactions between those chains, it renders the material insoluble, and instead it imbibes large amounts of water. It swells. Now, these classes of materials are very nice for embedding cells in. Here I'm showing some cells that I loaded up with a fluorescent dye so that you could see them. And so I can look at these cells embedded in here under the microscope. And this high water content is desirable because many times cells are secreting molecules to one another. And this is very important for how they communicate. So many different growth factors called chemokines and cytokines. Many times when I'm culturing these, either outside the body, or if I'm putting them inside, there are exogenous signals that I might want to deliver. And because of the high water content, those molecules can be transported very, very quickly. In fact, many of the soft tissues in our body have high water content. That also gives some very interesting mechanical properties to these materials. And we're learning more and more about how cells receive mechanical cues from this environment. So it becomes equally important to control the chemistry and the mechanics in this microenvironment. So then now some of the questions become, what materials should I use and what kind of signals do I need to give to these cells, and on what contextual way and time scales and spatial scales should they be delivered? Now, there are also many different types of hydrogels, and one that we happen to have worked with quite a bit are these polyethylene glycol hydrogels. So here's PEG, and one can imagine modifying the ends of PEGs with many different reactive groups. One of the early ones we worked on were just these acrylate end groups, where these macromolecules could be dissolved right in the presence of cells in a suspension. And then we would do different types of initiation reactions, many times light-based or redox-based, so that one wouldn't have to change the temperature or pH when you're doing reactions in the presence of cells. That's one of the main constraints. And so one could polymerize these chains and form a hydrogel network right in the presence of cells and encapsulate them. And PEG is nice for a couple of reasons. It's widely used in human medicine, used to modify many different types of proteins and small molecules to change their availability and their distribution in the body. And it's also one of the synthetic materials that when you put it in a complex mixture of proteins, again, whether that's in the body or outside the body when you're culturing cells, it resists a lot of non-specific absorption of those proteins. And that's important because if you want to understand how a cell receives a signal from outside, you don't want to have that signal that you're going to introduce blanketed by an absorption non-specifically of different proteins. So it's very useful from that perspective as well. But if you just use a PEG-based material, what happens is that cells don't recognize this chemistry. Proteins don't non-specifically absorb to this very well. So cells, when they're embedded in this type of hydrogel environment, they tend to take on a spherical morphology. And there are some cells in our body where this is very physiologic. So these happen to be the chondrocytes found in cartilage tissue, and this is the way that they look. And they build this lacunae around them, and they secrete and make cartilage matrix. So these gels can be very nice for these types of applications. It's also really good when you want to promote cell-cell interactions rather than cell material or cell matrix interactions. And so here's a cluster of the cells found in your pancreas, those ones that make insulin, the islets. So PEG gels can be very useful for this, but many, many cells in our body get information. They require information from this extracellular environment. And so here I'm showing you a picture of some cells found in your bone marrow that are capable of responding to injury and helping to repair bones and cartilage, and they also migrate throughout your body. And if you put these cells inside of a PEG gel, what happens is they want to spread, but they don't find anything to attach to. They're trying to move to one another, but they're inhibited by the interactions of the PEG gel, and they can't break it down with any of their enzymes that they know how to make. And instead, these cells, after a day or two, are going to undergo a programmed cell death called anoikis because of lack of interactions with that material environment. So this is then the perfect opportunity where we can begin to think about trying to consider these niches of adding in chemistries to try to make a material system that could better mimic what these cells would experience in their niche in the body. So we think about these matrices where first we have this blank slate of a PEG gel. And on the one hand, so my title was Cellular Control and a couple of clicks, we're interested in different types of click reactions where we can add in chemical functionality that the cells will recognize and learn specifically how cells respond to those signals. And we might want to do this in a way that's sequential, introduce one signal at one time and another at a different time, or spatially with gradients and different types of patterns. And so that's one of the reasons I'll tell you a little more about some of the photoclick reactions we've been interested in. But in cellular environments, it's not just about adding cues. Many times it's as important when those cues go away. And so we also have been interested in some complementary reactions. That's the removal of that signal, the cleavage of it, so-called photoclip reactions, where we introduce groups that contain linkers that can be cleaved upon exposure to light. And we can use this to not only remove chemistry, but also as a way to break down our material and control degradation in a spatially defined manner. So let's start over here with just some of these click reactions. And I'll give one more background slide, and then I'll give you an example of where we use some of this. So many of you in the audience, I've looked at some of the poster titles. I know that you work in this space and are doing some very creative work in designing different types of click reactions that can be used to conjugate different types of biological molecules. And that's really what we're interested in as well, is how can we click in biological molecules into this PEG-based background. And over the last decade or so, there's been an explosion of different types of chemical conjugation reactions that are considered bio-click reactions. So these are reactions that proceed with high specificity, oftentimes in a complex milieu of many different molecules. They can be controlled in a manner that allows them to be performed in living systems. And in particular, my group has been very interested in this thiolene bio-click reaction and have been involved in using this in biomaterial development. And the reason we're interested in that one is because it's one of the few click reactions that can be photochemically driven or photochemically initiated. So I'll tell you a tiny bit about this chemistry. But using this, what we're very interested in are designing these hydrogel precursors, these macromolecules, and then we put these N groups onto this so that we can click together our hydrogel networks where we can now begin to think about introducing biological functionality. So I'm going to walk you through, in the next 20 minutes or so, some different vignettes of how we use this. So let's start here, where we look at a tissue regeneration problem, where we're very interested in how can we help bones heal faster, especially when they're critical size, such as if you've had a major car accident or if you've had a tumor in your bone that's had to be removed. And so in doing that, I showed you that picture of those bone marrow cells that are very important in the healing process. So we began to become interested in how could we design something to promote bone regeneration by mimicking some key features that are found in the matrix that forms when a wound happens in our body. And so in this native tissue matrix, some of the key functionalities that are important is that there are adhesive proteins that the cells can bind to. Oftentimes they'll get survival cues from this. There are also linkers that can be degradable by enzymes or proteases secreted by the cells. And they decide on demand when to degrade and remodel this tissue. And then there are also different types of sequences that can serve as a reservoir for binding different growth factors, and they're released on demand by cells when it has to repair a tissue. So we began to think about trying to mimic or capture some of these basic functions into our PEG hydrogels. And in the biomaterials community, many people use peptides as mimics of some of these full proteins that are found in the native tissue matrix. So the reason that these peptides are nice is one can use an instrument, a solid phase peptide synthesizers, to make these short repeat units found in proteins. And we know, for example, on many adhesive proteins, we know the exact regions that cells bind to, such as this RDD sequence found in fibronectin and many different adhesive proteins. Here's a sequence that's found in collagen, and certain matrix metalloproteinases that these cells make can degrade this sequence very efficiently. And we can also pan through many different libraries of peptides and find ones that will bind some of the growth factors that we might want to use to stimulate our cells to make the bone repair faster. This one happens to bind to one of the BMP families that's involved with bone regeneration, TGF-beta. So the idea then is for our click reaction, we're going to use this thiolene reaction, which is photochemically driven, where the thiol part is simply the amino acid cysteine. So cysteine has a pendant thiol, so I can create and make various peptide sequences of interest and incorporate cysteine into that. And then our PEG molecule, the key part, is finding an ene that will react very efficiently with this thiol. But when you mix these two together, they're very stable and don't undergo a side reaction like a Michael addition that some of you might be familiar with. But these two instead, when you introduce light in the form of an initiator, undergo a thiolene step growth reaction, and it's radical mediated, and form these hydrogel networks. So the idea is I can mix in different peptide components and different PEG sizes and molecular weights to create these hydrogels. And if you combine this, now we have all kinds of robotic techniques where I can deliver nanoliter quantities of these hydrogel precursors along with the cells of interest, and I can screen through many different biomaterial formulations where I change the mechanical parts and properties of the hydrogel by the molecular weight of the PEG or the number of linkers or arms it has. And then I change the biological signals by the peptides that I synthesize. And I can synthesize those depending upon the cells of interest that I'm working with and the signals that I would like to. And then I can look under a real-time microscope and track and watch those cells and screen for formulations that lead to a desired biological output. And I'll just show you one example here. Let's take those same cells that we looked at before in the PEG gels, and now if I put them in this formulation that has these peptide components where it has a degradable linker and a linker that the cells can attach to, now we're looking at a low magnification of those cells. So these are all the cells, and they're spreading. They're locally degrading this matrix and creating tracks through it. And I can screen for formulations where I have high degrees of motility. And I can screen for formulations that also prevent these cells from migrating through. They can bind to the material, but they can't move through it. And so one can now begin to think about designing matrices that one can put into wound sites that can recruit local cells that are important for wound healing while keeping out other cells that might be causing scarring or undesirable side effects. And so this was one collaboration that involved an undergraduate student, a graduate student, and we had a medical fellow that came in that was from otolaryngology. Took me a while to be able to say that word, but these are basically surgeons that are interested in trying to treat craniofacial defects in your head and in the skull. So what we did was we created this critical size defect, 9mm defect in a rat skull, and we tried to repair that. And when we put in our hydrogel that the cells couldn't easily migrate through, we got very little healing, some around the edges here. But when we put in our hydrogel that served as a provisional matrix that could recruit some of those bone marrow cells, we could see large amounts of healing across this critical size gap. And so what was interesting and exciting about this is just material chemistry. We aren't delivering a drug, or we aren't delivering cells even at this stage. We're delivering a material to recruit local endogenous cells and influencing healing. So some of where this is now going is that I'll acknowledge, right, this isn't completely healed. It takes 9 weeks. And so this is where some of the synergies can be useful. So we can now learn about, do we need to have cells recruited to at a higher rate, so introduce chemotaxis or directed migration, or do we need to have more osteoinductive factors in this? So that's where some of this research is going. Now I'd like to take a few minutes and also talk, though, about the other side of this. So on the one hand, we can add in lots of functionality, peptides and proteins and hydrogel formulations. But in many cases, it's also important to remove that signal, or to be able to control not just the chemistry, but to control the mechanics of this. And so now I'd like to maybe put this in the context of one of the areas where we were really interested in not only how could we repair or regenerate tissues like bone and help them heal faster, but how could we regenerate tissue equivalents or understand how disease occurs and have disease models. Because if we could figure something out about disease, maybe we could find better ways to discover new drugs or ways to reverse disease where currently there aren't any drugs to do so. And so this is an area that we've been working in for the past 5 to 7 years or so, where we've been interested in cardiac fibrosis, scarring and stiffening of tissues in your heart, and one of those are your heart valves. So heart valve fibrosis. So here's a picture of a heart valve. Your heart has 4 valves. It directs flow within the chambers of your heart, coming in and out as well. It has to open and close 60 to 80 times a minute. Many times it's directing blood flow. There's a huge pressure drop across this. From an engineer's perspective, it's a perfect one-way valve that's supposed to last our entire life. But this valve here, so this is made up, it has these 3 leaflets that open and close with the beating of a heart. And these leaflets are about 300 microns thick, 300 to 400 microns thick. It's a very delicate tissue. And the cells that reside within that tissue are constantly undergoing an activation to go into repair mode when it's injured. And then once it's repaired and regenerated the leaflet, it goes back to a quiescent state. But sometimes that activation goes wrong. And it stays in this activated wound healing state. And when it's in that activated state, it's making tissue, it's depositing tissue. And what happens in that case is that all of a sudden you get a really thick leaflet. And when it gets really thick, it doesn't open and close very much. And your cardiovascular surgeon can see this happening and measure this, but they don't have good ways to reverse it or stop it. So we don't know what's going wrong, what's causing this activation or wound healing state. And you can see that in this picture here. If I take a thin section of this valve leaflet and I make it into a thin section and I stain it, I can put it under a microscope and look at the cells within this leaflet. And one of the things I can stain for is this protein called alpha smooth muscle actin in the red here. That is the activated, the wound healing valve cell. And in healthy tissues, you only have a small amount that are activated at any time. But in diseased tissues, you have lots of these activated cells called myofibroblast. So we got interested. We wanted to understand what causes this activation. And if we could understand what causes this activation, could we design drugs to inhibit it and reverse this? So that was the challenge of the field, and we were interested in studying this. And we were collaborating with some molecular cell developmental biologists. And so we were set out, we had some ideas of what was causing this, and so the student at the time was a biologist that we were jointly advising. And we would take these valve cells outside of that tissue and we were going to study them And the classic way to study them is you put them on a tissue culture dish, which is made of polystyrene. And the thing about these valve cells is the minute you take them out of the tissue and you put that on that stiff polystyrene surface, they all become these activated cells that have this alpha smooth muscle actin. I'm going to call it a myofibroblast. And look at this little video. The minute you take it out of the tissue, they all become these myofibroblasts, these wound-healing cells. They're making this protein. They're highly contractile. They even snap together and form these nodules that will eventually even calcify in the in vitro tissue dish. So this could be a good model if you wanted to study disease progression in heart valve cells and cardiac tissue. But the problem is that there is no way to study what are the cells really like in their quiescent state, and could I study the process of them activating and then how they deactivate. So this molecular biology student started collaborating with a material science and one of my chemical engineering PhD students, and we were already doing work with these hydrogels. And so the first natural question is, well, is part of this because tissue culture polystyrene is a gigapascal in stiffness, 6 orders of magnitude higher than what a soft heart valve is. And so we put some of these cells into our hydrogel systems, and lo and behold, when we matched the gel mechanics and had cultured these cells in soft gels, we could begin to culture these cells like their quiescent fibroblast state in the native tissue, and not like the activated myofibroblast and tissue culture polystyrene. And it resembles very closely, and you look at a valve section, so very few activated cells. So that was our starting point. And then we got interested in, so if we can culture these cells in this quiescent state, now we can begin to ask questions. What's the role of how these cells sense that mechanical signaling? And is that really important in disease progression? And if I could figure out how they sense mechanical cues, could I inhibit that and use that as a target to try to reverse heart fibrosis? So we could ask this hypothesis, but what we wanted now was we wanted a material system where we could not just control mechanics and control chemistry, but we wanted to dynamically change it. And so the student working on this was designing some materials where we could use light to break linkers inside of the gel. So here's our PEG molecule, and now we insert a chemical group, this nitrobenzyl ether, that when you expose it to light, it will undergo an intramolecular cleavage reaction. And so if I put this into my network and I connect up my hydrogel with these linkers that are light sensitive, then when I expose them to light, I can begin to start breaking down my hydrogel links, and that's going to influence the mechanical properties of the environment. And so we use, this is a rheometer that applies an oscillating stress to a hydrogel, and it's really sensitive to the links inside of this hydrogel material. And so when I expose to light, I'm going to change the mechanics, and I can in situ monitor that with this rheometer, where I measure the modulus, or somewhat the stiffness of the material, as a function of when I expose it. And I start out with a stiff material that I can then soften with exposure to light. And one can do this using different wavelengths of light and intensity, but I think the interesting part is right here. So now I can have a cell-laden material. Watch those cells when they're in a quiescent state or an activated state, and expose to light and change the mechanical environment and watch how the cells respond. So I expose to light and shutter the light source, and now I have material that has half of that modulus. I've softened it. And then when I remove the light source, it's completely stable at that new steady state. And then I can re-expose again and watch how the cells respond. So we did that. So we put the cells in these types of environments, and we found regions where here the cells were still activated, but that if I softened it to here or to here, they would deactivate. And we could watch that deactivation in time. And we could quantify that, and we could ask questions. What happens when cells are in soft environments where they're deactivated and stiff? And can I go back and forth between these states by dynamic changes in the mechanical modulus. So why is this important? So our biology friends, when we do these types of experiments, they can look at all the intracellular pathways that are going up and down when you're making a material that's stiff or soft. And so I won't go into the details of this, but if one knows that entire network, then you can find certain nodes that are highly going up or down. And once we know that, we've been able to use that to design different types of hydrogel systems that can deliver inhibitors to those nodes. And now we can take heart cells that are normally activated, and if we treat them with those inhibitors, we can make them quiescent again. And so we're collaborating with clinicians at looking at different ways that we can deliver these molecules to targeted regions and tissues to try to inhibit and use it as a treatment to try to reverse these types of fibrotic disease. All right, so my time is quickly running out. But what I'll tell you is just one small final example then is another way you can kind of think about this is we've designed systems that mimic the native extracellular matrix. And when we do experiments with that, we're being observers of cells. We're trying to design systems that match the in vivo environment in vitro, and we can learn something. But then we also, with some of these light-triggered chemistries, we can be active experimenters. And I don't just have to passively observe. I can introduce chemistry, and I can change the mechanical environment in situ and watch how cells respond to that. And that can be really useful when one is trying to understand and control very complex systems, such as many of the stem cells that one works with, or if you're culturing multiple different cell types. And this is where the photochemistry really is valuable because I can pattern in chemistry into regions of hydrogels by exposing through mass and controlling illumination of light. I can use 2-photon confocal microscopy, not just for imaging my cells, but also for introducing chemistry. If I combine that thiolene reaction to add things in with the cleavage reaction to remove, I can create gradient structures and then cleave away certain regions of that gradient. Many times biology is about opposing gradients or waves of molecules. And so this is what we think about. We think of cell culture niches where we can expand and look at stem cells, add in chemistry and different signals at certain regions in time, sequentially add in a different signal at a different point in time, and then I can look for regions of where do I see proliferation that's high if I want to expand those cells. Or perhaps not just high proliferation, but I look for regions where the cells are starting to become more like bone cells or osteogenic. And not only do I watch these cells, but if I use the photocleavable chemistry, I can capture cells in a certain region of interest and then gather those particular cells that I'd like and do further analysis or deliver them in vivo. And so with that, I think I'm just going to skip to my summary. And I hope what I've shown you is there's lots of interesting ways that material chemistry and chemistry reactions are enabling the field in biomaterial science and biology and in clinical medicine to address really critical problems for our field. Dynamic environments are very important. I think they allow us to answer really interesting questions about how cells get signals from their environment and how they can be used to learn different ways to deliver molecules. And when you integrate these, you're able to design really complex culture systems that I think help serve as a bridge between traditional in vitro culture and in vivo experiments. And with that, certainly I'd like to acknowledge my group members. I think that's another thing about the Dreyfus Foundation is that this truly isn't just about doing great science, but it's about the people that we get to train that do the great science. So everything that I showed you was effort of former and current lab members, and I thank you all for your attention.
Thanks a lot. I think you've really given vivid examples of what we mean by highly controlled. We want to have some questions, so please. You hold on a second. Yeah. Here you go.
Oh, that was a really amazing talk. Thank you. I'm wondering, when you do these hydrogel experiments, how many cells, in the images it looked like there were just 1 or 2 cells, but you were zooming in on small regions. But what sort of number of cells are you using, and what's the heterogeneity among the hydrogels?
Yeah, so the cell density is something that can be controlled. We've gone as high as like 40 million cells per milliliter or cubic centimeter, which is a pretty high density, all the way down to something that's very low, like 250,000 cells in that same environment. So it kind of depends. Sometimes if you want a tissue model, you match the tissue. So cartilage is about 5 million cells. But if we really want to study cell matrix interactions, sometimes we make it really dilute so we don't have to worry about cell-cell communication, so we intentionally put the cells very far apart. But that's something you just control right when you're encapsulating the cell. What was the second part? Was that? Heterogeneity. Heterogeneity. So the hydrogels themselves, we haven't intentionally introduced much heterogeneity. That on the size of a cell, which is like 10 microns. There's certainly heterogeneity and cross-linking density in chemistry on a molecular scale, but over a cell that integrates over all that, it's fairly homogeneous. One could envision going with graded structures that are more reminiscent of tissues as well. A lot of tissue has hierarchical structure to that as you go across a bone or a piece of cartilage.
Following up on the heterogeneity question, are the PEG hydrogels stiff enough that you could ablate microfluidic channels in them and actually run fluids through them without having to introduce yet another material to channelize things?
Yes, so we've done some of that with using 2-photon confocal microscopy. Create a monolithic hydrogel where one can go in and write channels or road features. But the high throughput of it isn't as nice as one might like, as the size of the focal plane and the 2-photon. So you make smaller features and smaller devices.
Very cool.